Welcome back, anatomy students. Today is Wednesday, and we are continuing in our cardiology system study with blood types today. And as I said in the previous video, for many of you, blood typing should be a review, especially for those of you who completed the Apologia curriculum for biology last year, because this topic was covered fairly in depth. But for those of you who maybe didn't do that, or this is a review, we are going to go over some blood type information. All right, and so you guys probably know that there are 4 blood types in humans. That's A, B, AB, and O. And the difference between the blood types lies in which antigens are on the surface of the erythrocytes, or the red blood cells. An antigen can be defined as a protein or other molecule that, when introduced into the body, triggers the production of an antibody. And so if you recall, your body produces antibodies as a way of fighting off invaders. And so therefore, we could say that antigens trigger an immune response. All right, and so what we just said is the difference is what antigens are on the surface. So if you look here, type A is going to contain A antigens on the surface. Type B is going to contain B antigens on the surface. Type AB, you guessed it, is going to have both A and B antigens on the surface. And type O has neither A nor B antigens on the surface. So it's the presence of these antigens that dictates which blood type you inherited. Okay? And so why do blood types matter then? So antibodies in your blood are produced to fight infection. As we said, they're sent out as a defense mechanism. And so if you're ever given the wrong type of blood, those antibodies are going to attack the antigens on the surface of the wrong blood cells and build what we would consider bridges from one red blood cell or erythrocyte to another. And you don't want that. You don't want blood cells building bridges connecting to each other, because we know that they need to be flowing freely through our blood vessels to get where they need to go. And so if they form bridges, they clump, and this is very serious and very bad. It blocks the flow to organs. And so if we look here at the following chart, it's going to show you which antigen is present, if any, and which antibodies are produced, if any. Okay, so if you have erythrocytes in your blood, type A antigen, the antibodies in the blood will be antibody against the B antigen. If you have type B blood, you have that B antigen on your surface, so the antibody against the A antigen will be in your blood. If you have type AB, you have both A and B antigens on your blood cells, and so there's no antibody in the blood. And type O, you have the O antigen, and you have antibodies against both A and B in your blood. Okay? And so antibodies, however, are not naturally present in the blood. They are only synthesized or made by your body when they are needed. So, when would they be needed? In the unfortunate exposure to the wrong blood type. And this is very rare. Medical, at this point, has come a very long way to ensure that the donated blood is typed correctly and you get the blood that you need. But, you know, there are rare circumstances in which you would be given the wrong type of blood, and that would not be very good. Now, this whole concept is different for a baby that is 2 months old or less. The antibodies will appear in the body if the baby's blood type allows for the production of 1 or 2 of these antibodies. And we're talking, again, 2 months old or less, so just the first 2 months of life. Okay? Food or other organisms like bacteria can carry the A or the B antigens, and the intestines come in contact with these antigens, and it is just a short matter of time before everyone has been exposed to both A and B antigens as a young child. And as long as your blood type allows for the production of antibodies, they will be made by your body. You probably have heard this before as well, but type O is considered the universal donor. There are no A or B antigens on O erythrocytes. Therefore, the antibodies cannot produce an attack. Okay, and that is why O can be considered the universal donor. Okay, and so despite being the universal donor, it's preferred still to receive a transfusion from someone with the same blood type. So yes, you can receive from an O because of the lack of those antibodies. However, it would still be preferable to receive blood from someone who matches your type exactly. But type O, like I said, can be used in an emergency situation. So you may be asking yourself or thinking back to biology from last year, but what determines blood type? So how did you get your blood type versus your brother, your sister, and your mom and dad? Well, it's genetics. And so if you studied last year, the allele for type O is recessive, and the alleles for types A and B are both dominant over O. However, important to note that A and B types are not dominant over each other. So we wouldn't say that A is more dominant and B is recessive. They kind of negate each other. They cancel each other out, or they exhibit what's called co-dominance. So if you were to get an A allele from mom and an O allele from dad, again, we can say that O is recessive and A and B are dominant over O. So you'll have type A blood because it's dominant over O. If, however, you get an A allele from mom and a B allele from dad, you will get type AB blood because neither is dominant over each other. They exhibit that co-dominance, and so you will have AB blood. And so this is just one example from your book. It's actually an example from the Apology of Biology book from last year, if you used that book. And I'm not going to go through this in this video, but just make sure that you understand and review the concept of genetics through the Punnett square that you should have covered in a biology class. And just make sure that you understand how this is working and what's dominant or co-dominant over another. I did throw in just a brief little bit of humor for those of you, I'm thinking Gabe, who I know is a caffeine addict. He always has his coffee. But no, Mr. Vance, AB caffeinated is not a blood type. Ha ha ha, I know, I'm so corny. Continuing on with blood types, guys. So, an AB antigen on an erythrocyte is very important. But, as you guys know, when you talk about blood types, we usually indicate if someone is positive or negative for something. And so that brings in the next factor that we have to consider when we talk about blood types. We're talking about the Rh antigen, which stands for the rhesus monkey, this little guy right here. He's cute. And so this was where this antigen was first discovered, in the rhesus monkey, and so we call it the Rh antigen. And if you have the Rh antigen on the surface of your blood, we would say that your blood is Rh positive. That's where you get that positive after your blood type. And if you have no Rh antigen on the surface, we would say you are Rh negative, or you have that negative. And so about 85% of the human population is Rh positive. So the vast majority of us are O, A, B, or AB positive. So the negative, having a negative blood type, would be considered fairly rare. And the Rh antigen is super important. It's as important as a person's blood type in terms of A, B, and O, and AB. So it is important to know whether someone is positive or negative. And so this also, like your blood type, AB, A, B, or O, is inherited via genetics from a gene. And it's separate, though, from the ABO type. So this gene is completely separate from whether you're A, B, AB, or O. In this case, Rh positive is dominant, which hopefully, if you remember anything from learning genetics, that makes sense because I just said 85% of the people in the world are positive. So likely that's going to be a dominant gene, whereas Rh negative is a recessive gene. So if you have just one allele for Rh positive, then you will be Rh positive. So you only need to get one allele from one parent to be that, whereas to be recessive or Rh negative, both mom and dad would both have to be Rh negative for you to get that, which makes it more rare to have that combination. All right, so the antibodies can be produced against an Rh antigen. So people who are Rh negative will produce antibodies against the Rh antigen. The Rh negative person has to be exposed to Rh positive blood in order to produce that antibody. And so this can happen through a blood transfusion if you need a bag of blood, or this can happen in pregnancy. And typically in textbooks, we talk a lot about the Rh antigen in terms of pregnancy because there are some risks to mom or baby because of the existence of this antigen on our blood cells. If the mother is Rh negative, again, this is more rare, there may be a problem if she has children who are Rh positive. And again, that's determined by a completely separate allele from their blood type, ABO. But depending on the genetics of mom and dad, if they were to be Rh positive children and she's Rh negative, there could be problems at birth. During pregnancy, if you didn't know this, the baby's blood doesn't necessarily mix with the mother's blood. They're completely separated by the placenta. So the problem doesn't lie during the pregnancy. It actually lies during the birthing process. So the Rh antigen would be foreign to the mother in this case, okay, because she's Rh negative. So her body has never been exposed to the Rh positive antigen. So her body will therefore make anti-Rh antibodies. And this actually is harmless for the first baby that she has. So if it's her first pregnancy and she's Rh negative and her baby comes out Rh positive, it doesn't make a difference. That baby and mom will be healthy at that point. And it's harmless to both mom and to baby. However, the problem arises if that same mother who's Rh negative has a second child who is Rh positive. This puts the baby at risk. And it puts the baby at risk for a disease called the hemolytic disease of the newborn. And so how does this work? Well, during pregnancy, the anti-Rh antibodies of the mother will cross the placenta. These antibodies attack the unborn baby's Rh positive erythrocytes. Because remember, mom is Rh negative in this case, baby is Rh positive. Okay, and so now we're attacking the Rh positive antibodies. And this can either have a really small effect on the baby or a very big effect. It kind of varies from a person to person case. But the more Rh positive children that this particular Rh mother has, the higher the risk for each baby because of this exposure. Okay, and so fortunately, because of advances in medical, we know that we can treat hemolytic disease of the newborn. And so Rh positive newborn, okay, from an Rh negative mother, will be given a transfusion of its ABO blood type, except that the donated blood is Rh negative. Okay, so see that distinction there. They're giving them the opposite. Instead of positive, they're giving them negative. So if a baby is A+, the baby would be given A-. Okay, what happens here is the negative cells replace the positive cells. Then the anti-rich antibodies from the mother will gradually break down, and they will not make any more because he or she is Rh positive. And when we do this, the baby is able to receive the oxygen-carrying red blood cells that it needs. So that's how we treat it, but more commonly, hemolytic disease of the newborn is completely preventable in the first place, and we never have a reason to treat it by giving the baby the opposite type of blood, Rh antigen. So typically, it's prevented. Soon after a baby is born to an Rh mother, the umbilical cord of the baby is tested, and if Rh negative, then they don't need to do any treatment. But if the baby is found to be Rh positive, then they need to do treatment. The mother in this case, after having this first child, will be injected with anti-Rh antibodies, which will quickly attack the Rh plus cells that they have entered that may have entered into the mother's blood during childbirth, and the mother's immune system will not be stimulated to make her own long-lasting production of Rh antibodies. Okay, and so this is commonly how they will treat newborn mothers after their firstborn child. And this is usually something that doctors are testing before you even deliver a baby. They usually want to know what the mom is versus what the baby is. And that's a standard pretty medical test that gets done. All right, and so this is just the image from your book, just to show you, for reference sake, that the first pregnancy, we're fine. There's no mixing here. Here's the baby, here's the mom. But if blood mixes at delivery, you have pluses and minuses that get mixed, and the mother will start to produce the antibodies to the Rh plus because she's Rh negative. Any subsequent pregnancy with the child now is going to have destructive antibodies which cause issues to the blood cells. Okay? And so again, keeping in mind that about 85% of women are Rh positive, so this is only a small percentage of risk for most women. Plus, if an Rh woman has if an Rh negative woman has Rh negative children, there is no risk. And so even pregnancies can, subsequent pregnancies may not pose a risk at all if all of the offspring end up being Rh negative. It only becomes a problem if you have an Rh negative mother with an Rh positive baby after the first birth. Okay? And that is going to be the last slide that we cover in this video. However, I have two very important videos that I really would like you to watch, so please make sure you watch those so that you better understand the genetics and hemolytic disease of the newborn and the Rh antigen. All right, so those videos will be linked below. Please just make sure you take those 5 or 6 minutes to watch those after you finish this video, and I will see you again on Friday for our last remote learning video for the week. All right, see you then. Bye.